This is lecture 6 and we'll be going over the MHC. The objectives for this lecture is that we're going to be going over the MHC expression, the composition, classes, and the different roles that the MHC plays in adaptive immunity. And later in the lecture we'll be going over paternity testing with HLAs, and we'll do a couple exercises to get you familiar with paternity testing. A couple of definitions that you will need to know if you've taken a general genetics course, then these are kind of just refreshers. If you have not, then please know each one of these definitions. Chromosomes, obviously, they're the strands of our DNA that carry all of our genes, and we have 23 pairs of chromosomes in each human cell. A gene is a unit of genetic material that codes for hereditary traits, so our eye color, hair color, skin tone, etc. Locus is actually the position of that particular gene on the chromosome. An allele is a different form of a gene that codes for a slightly different form of the same product. Haplotype, these are a set of genes that are located close together on the chromosome. And genetic polymorphism, we consider the MHC a polymorphic gene cluster. It's a variability at a gene locus in which the variants occur at a frequency of greater than 1%. So MHC is one of the most polymorphic gene clusters in humans, so it has great variability. So this picture just basically kind of shows what the MHC does in a nutshell. So we have our APCs, or antigen presenting cells, and they present antigens to our T cells, naive T cells. Remember, our T cells have the TCR, or T cell receptor, and so the APC will present this antigen through the MHC2 molecule to the TCR, and if the T cell recognizes this antigen, then the T cell will become activated and it will start to proliferate and differentiate. And we'll deeply go into this interaction in this lecture. So MHC molecules are found on all nucleated cells. So if I ask you, do mature red blood cells have or express MHC molecules? The answer would be no, because they are not nucleated. What about a nucleated red blood cell? They have been found to have small traces of the MHC molecule, class I. So in this instance, then yes, they do contain small traces of MHC molecules. But going back, so when we say all nucleated cells, we're mainly talking about our white blood cells. MHC molecules play a huge, enormous role in the humoral and cell-mediated immunity, and their main function is to present antigens to our T cells to get them to recognize these potentially dangerous antigens. The MHC molecules also have been linked to involvement of transplants, graft rejections, and also other autoimmune diseases. So we'll kind of take a look at these as we go through this lecture. A little history about the MHC, or the discovery of the MHC. Dr. Jean Dausset won the Nobel Prize in 1980 for the discovery of the HLA antigens. He observed an anti-leukocyte agglutening substance and later published on the relationship between leukocyte compatibility and antibodies response to skin grafts. So he played a major role in the discovery of the MHC molecules, along with two other scientists. So our HLA antigens are our MHC molecules. So the genomic sequences that are the code to make HLA antigens are called MHCs. And these genes are located on chromosome 6. And we have three different classes of MHC. We have class 1, which is located on loci A, B, C. We have class 2, which is on loci DP, DQ, and DR. And then we have our class 3, which are complement and cytokine formation. So this makes up our different components of our complement and like tumor necrosis factor, etc. We'll be mainly focusing on class 1 and class 2 during this lecture. Here's just a simple karyotype if you've seen one of these before. Here I was just wanting to show you chromosome 6, and this is where our HLA genes are found. Again, on this tiny little region on our chromosome 6, we have our class 1, class 2, and class 3 HLA gene complex. So here is where all our information is stored for our class 1 and class 2 MHC molecules, and our class 3 as well. But we're mainly going to be focusing on our class 1 and class 2. So we have our class 1 and class 2 MHC molecules. They're basically the same, however their chemical makeup is different, which we will go over in the next couple of slides. But as a whole, basically it's like a hot dog in a bun. Okay, we have our antigen in between the bread, and our bread is our MHC class 1 or class 2 molecules, and it's going to present this peptide or antigen to our T cells in order for the T cell to recognize it, or see if the T cell recognizes it in order to stimulate that T cell. So let's take a look at our structures of our class 1 and class 2 MHCs. So you can look on this diagram, and where it has the arrows pointing to the peptide binding cleft, this is actually where the antigen is stored to be presented to the T cell. So let's look at some common differences between the class 1 and class 2. So class 1 has an alpha 1, alpha 2, and alpha 3, and in the bottom right it has this beta 2 microglobulin. This particular protein will actually has many benefits and plays certain roles in certain diseases that we will discuss later on in this class. Class 2 molecule has an alpha 1 and alpha 2 and a beta 1 and beta 2. You will not need to label these on an exam or anything. This is just help you to visualize the class 1 and class 2 MHC molecules. So let's take a quick look at the allele diversity of the MHC. So we know that alleles are alternate forms of a gene that code slightly different varieties of the same product. Okay, so remember that at the beginning of the lecture I said that the M8 system is one of the most polymorphic systems in the human body. So we could take a look at Class I HLAs, and we have HLA A, B, and C. So we have 2,000 currently, as of today, we have 2,013 different alleles for HLA A. For B, we have over 2,600 different alleles. And for HLA-C, we have over 1,500 different alleles. So if you think about it, we have, I believe the correct number is like 1.7 billion different possibilities for possible allele variants. So this is highly, highly polymorphic, and immense variability for this MHC molecule. And this chart is just taken out of the second edition of your book. I believe you should have the fourth edition, so this is fairly old. But you do not have to know this for the exam. I just wanted to give you a little insight on the different HLA specificities for each particular HLA. So we have A's, B, C, DR, DQ, and DP. But there are multiples of new HLAs that has been found since then. So needless to say that our allele arrangement is highly, highly unique. We could almost make it like our fingerprint. Alright, so the good news is that we could use this to determine paternity testing or maternity testing. The bad is that it could usually create problems when we do transplant. So because our body's not recognizing these new molecules that have been introduced into our bodies. So this is why we have issues with rejection, transplant rejections or graft rejections, etc. And we'll have a whole lecture going over graft rejections, etc. So for now, so each person has 2 alleles for each HLA location. Okay, so we inherit 1 allele from our dad and 1 allele from our mom. Alright, so if we have different alleles at one locus, so we, as an example, A10 and A43, so we inherited A10 from our dad and A43 from our mom, then we would consider this to be an heterozygous allele. Alright, but if we have the same allele at the A locus, alright, we have A36 from our dad and A36 from our mom, then we'd consider this to be homozygous. This is highly unlikely, but it has happened. It's a very small chance. So our Class I MHC, all of our nucleated cells have Class I MHC. Okay, we can kind of almost consider this as an ID for those nucleated cells. Alright, so depending on what type of cell it is will determine on how much the molecule is displayed. Alright, so lymphocytes have many Class I MHCs on its cell surface, but our liver cells only just has a few. Alright, so depending on what type of cell it is will determine how much of the Class I MHC is displayed or expressed. So our HLA-C antigens are expressed at lower levels, than our A and B antigens as well. So something to think about. When we are doing a transplant, we want to make sure that we have HLAs that typically match. If we have an exact match, that would be obviously ideal. But since our MHCs have a wide range of variability, it's very hard to find that perfect match. Alright, so, and depending on the transplant procedure, certain HLAs may be more important than others. Remember that C are not expressed as much as A and B. So, for instance, in a kidney transplant, we want to have HLA A and B to be exact, and C, if it matches, that's great. If it doesn't, then they usually consider this as optional. So HLA A and B are mandatory, and HLA C are optional. Our bone marrow, obviously, this is starting from the ground up. We are totally replacing our hematopoietic stem cells. So we want to have an exact HLA match. All right. If you do not have this, then you run a huge risk of getting graft versus host disease. And I'm sure Cherika will be going over this in hematology, and we'll briefly touch on it later in this course. Our Class I MHC is composed of 4 glycoproteins, the basic 4 glycoproteins. We have 3 alpha proteins, so that's our alpha 1, alpha 2, and alpha 3. And then we have, remember, we have that 1 beta 2 microglobulin protein. Alright, now, as I told you before, this beta 2 microglobulin protein is used to detect certain diseases. The main one would be multiple myeloma, so we could use beta 2 microglobulin as a tumor marker for multiple myeloma. We usually use this marker as a determinant of the prognosis of multiple myeloma, and when we see increased levels, typically this determines that this patient has a poor prognosis for not recovering from multiple myeloma. And this is a simple test. We can detect this in our blood samples, so simple test to run. So what are the roles of the Class I MHC? It's highly involved in the antigen presentation to CD8 T cells, or our T cytotoxic lymphocytes. Alright, and one thing that we have to remember when this Class I MHC presents these antigens to these CD8 T cells, okay, is that this antigen has to be processed first. Alright, so when we think about antigen processing, we think about from within the cell to be pushed out into the membrane. So we basically call this an endogenous antigen because it comes from within. Alright, so this antigen has to be processed, and then the conversion of these proteins or this antigen is then pushed out and to the cell membrane in order for the CD8 T cell to recognize it. Alright. So this antigen processing for this Class I MHC is constantly being done and within the cell. So it's this MHC is constantly pushing out to the cell surface and showing these proteins, all right? And the T cells are looking for non-self antigens or proteins, which can be hiding within the cell, either from viruses, microbes, tumors, even certain parasites can be hiding within our cells. And if they happen to catch one of these, let's say a virus or what have you, and it gets pushed out, one of the proteins on the virus gets pushed out and the T cell recognizes it, then it will be triggered and it will actually kill that particular cell because it's not a self protein. You know, it happened to detect this virus's protein, and then in a way to protect the body, it'll make that cell go through apoptosis and destroy that particular cell. This is just a diagram of the Class I MHC and its antigen processing. So it is constantly going through this process and presenting new antigens with the Class I MHC on the outside of the cell membrane. So now let's talk about the Class II MHC. The Class II MHC can only be found on antigen-presenting cells, including monocytes, macrophages, dendritic cells, and our B cells, our activated T lymphocytes, and our endothelial cells. All right. So the Class II MHC is highly involved in antigen recognition by the CD4 T helper cell. The composition of our Class II MHC is basically composed of some polypeptides bound non-covalently with 2 alpha proteins and 2 beta proteins. By it being or having these 2 alpha proteins and 2 beta proteins, this allows for longer peptides to be presented to our CD4 T helper cells. So remember that our antigen presenting cells, or our APCs, play a major role in our Class II MHC. So our APCs are going to engulf these antigens and process these antigens and present these processed proteins to our T helper cells, or our CD4 T cells. All right. This will allow the alert of our T helper cells in order to activate these T helper cells to help stimulate our other immune cells to say, hey, there's something going on. You need to start dividing, multiplying, and producing antibodies to this specific antigen that I have detected. All right. So it basically aids in protecting us as humans from extracellular invaders, and we consider the role of our Class II MHC to be considered as these exogenous antigens. Okay, so outside antigens. And here is a little diagram representing the APC processing these antigens. So obviously at the very bottom you see the antigen is getting engulfed, and then it will be put onto the MHC Class II and then pushed out onto the outer surface of the membrane in order for it to present to our CD4 T cells. So this diagram shows each Class MHC pathway. So on the top we have our Class II MHC pathway. So we go through endocytosis of an extracellular microorganism. It's going to get processed, and then we, on our Class II MHC, after it comes out of the endoplasmic reticulum, it has this invariant chain on it, and once it actually binds with that endocytic vesicle with the processed antigen, it will lose that invariant chain, and then it will then be able to allow that antigen to fit into the Class II MHC, and then it will get pushed out to the cell membrane. So a CD4 T cell will be able to detect that particular antigen. On the bottom we have our Class I MHC, so remember that it is endogenous. Alright, so that means an antigen comes from within the cell, alright, like a virus, etc. So this antigen will get processed within the cell and get connected to a Class I MHC, and then it will be pushed out to the outer membrane or cell membrane in order for the CD8 T cell to detect this. Now, one thing to remember is that our Class II MHC pathway cannot present to a CD8 T cell, and our Class I cannot present to a CD4 T cell. So one way to remember this is that our Class II and our CD4 go together, so 2 times 4 is 8. Alright, and our Class I can only present to our CD8 T cell, so 1 times 8 is 8. So that's one way to be able to keep these two pathways, or these two MHCs, correct, by you want the value to be 8 total. So 2 times 4 is 8, so Class II and CD4, and 1 times 8 is 8. So we have our Class I and CD8. So here's a quick comparison of both our MHC Class I and Class II. Remember that our Class I has those 3 alpha proteins and that 1 beta 2 microglobulin. Remember, we use that beta 2 microglobulin as a to help determine the prognosis of people with multiple myeloma. Increased levels of this beta 2 microglobulin determines a poor prognosis for these patients. All nucleated cells have this Class I MHC, and we get these antigens or peptides from intracellular pathogens. So pathogens that invade the cell will typically be presented on the Class I MHC. And remember that these can only be presented to our CD8 T lymphocytes, our CD8 T cells. Our Class II is made up of 2 alpha proteins and 2 beta proteins. And remember that APCs and activated T lymphocytes and endothelial cells are the only ones that express this Class II MHC. And these peptides or antigens are derived from extracellular pathogens, so they have to be engulfed or phagocytized and processed, and then they get pushed out on the MHC Class II molecule. And these can only be presented to our CD4 T cells. When we're talking about HLA and transplants, this is probably one of the biggest barriers that doctors come across, because we want a successful allogenic organ transplant. So especially when we're talking about bone marrows, HLA, especially Class II MHC compatibility, is so crucial in trying to match because we have to have an exact match in order for this bone marrow to work properly, and so our body will not start attacking these particular cells because it's foreign, right? So typically when we do bone marrow, usually it's from a direct family member, a mom or dad, or a sibling. So typically when we do bone marrow transplants, it's usually from a close relative. Now, some transplants don't require any HLA antigens because usually they're like in an avascular site, like a cornea. So when you have cornea transplants, they will not do any HLA matching because it's not really right near any vascular site, so it doesn't have blood running directly to it, so we don't have to worry so much about it being rejected. And HLAs also play a big role in autoimmune diseases as well. One of the biggest autoimmunities that we usually talk about in immunology is ankylosing spondylitis, and the allele B27 is associated with this ankylosing spondylitis. And ankylosing spondylitis is basically inflammation of the spine. 70-90% of patients will have this B27 allele, but only 1% of that 70-90% will actually get that particular disease. So it's common, but it's not common, I guess, is one way to decipher that. So typically in a healthy spine, we have our disc in between our vertebra and our backs. In ankylosing spondylitis, the front and the back typically will eventually fuse, and creating what they call a bamboo spine. So those joints will get inflamed and will actually start to erode away the disc, and the bones will start to fuse with each other. So here's a quick review once you answer these questions, and we'll go over the answers in just a moment. So what is the function of our MHC molecules? It's to bind peptides within cells and transport to the cell membrane to be recognized by our T cells. Origin of peptides for the MHC class I molecules. Remember, these are intracellular or endogenous, so they come from within the cell. Our MHC class II are exogenous or extracellular, and remember that these antigens or peptides have to be engulfed and then processed and then pushed back out to the cell membrane. So which cells are considered antigen presenting cells? Are B lymphocytes, monocytes, macrophages, and our dendritic cells. And define haplotype. It is genes inherited as a set or combination of genes. Here's a chart. Why don't you go ahead and fill this chart in or write it down on a piece of paper, and we'll go over it in the next slide. Alright, so class I MHC molecule, the main gene loci is A, B, and C for class I. They are found on all nucleated cells, and they present the antigen to our CD8 T cells. Class II is DR, DQ, and DP. Remember, these can only be found on our APCs, activated T cells, and endothelium cells, and these present the antigen to our T4 CD4 T helper cells. So we know that alleles are alternate forms of the same gene. So a couple of examples would be our HLA-A alleles would be A1, A2, A3, A5, etc. B alleles would be B7, B5, 8, 27, etc., etc. Now, on our haplotype, remember these are a collection of alleles on the MHC genes that present on a single chromosome. So, for instance, person A, on chromosome 1, they would have A1, B5, CW1, DR1, DQW1, DPW1. This would be inherited from their mother or father. And on the second chromosome, we'd have A2, B7, CW2, so on and so forth. And then this would be inherited from either their mother or father. All right. So one thing to keep in mind as we go further into this lecture, remember alleles, they're alternate forms of a gene, and then our haplotypes are the collection of those alleles. Our phenotypes, so when we have our phenotypes presented, we know what types of HLAs there are, but not haplotypes. So we don't know the correct sequence. So for instance, in our class 1, we know we have A1, A2, B5, B7, CW1, and CW2, but we don't know which ones go with which, right? In our class 2, we could have DR1, DR2, and so on and so forth. When we find out the genotype, these are the actual alleles for a particular trait that are inherited, so we know the grouping of these. So we know the haplotype. So we have our A1. We'll find out that our A1, B5, CW1, DR1, DQW1, and DPW1 will be inherited from, let's say, our father. And then our A2, B7, CW2, so on and so forth, we have inherited from our mother. So our genotype, they are actually grouped together, and our phenotype, we have our HLAs, our known HLAs, but we don't know what sequence they are in. So how do we detect our HLAs, right? So the most common method is a microcytotoxicity assay. Usually we will use this particular assay to detect our Class I MHCs. Basically, we draw our patient's blood, and then we'll segregate the patient's lymphocytes, and then we'll add our known HLA antiserum, and then add that, and if it binds to the patient's cells, then we know that they have that particular allele or HLA. And if we do not see any color reaction, etc., then we know that they are not positive for that particular, in this instance, that A1 HLA allele. Most current method, obviously, is PCR. PCR is the up-and-coming testing platform that most labs are using now. This is a great way to detect our Class II and potentially our Class I HLA alleles as well. But it is always good to know about that microcytotoxicity assay because machines break down. So you always need to have a backup method, and that microcytotoxicity assay is a backup to the PCR. So when we're trying to determine HLA types, we'll basically get the sample from the patient and we'll process it. And this testing will help us determine the phenotype of that particular patient. So basically which HLA types are present in the patient's cells. Obviously, if we have two different types of one particular HLA, like A2 and A9, then we would consider them to be heterozygous. If they have A2 and A2, then remember that they would be homozygous or the same. By comparing these phenotypes from the child and mother or parental phenotypes, it can really help determine the possible inheritance of certain HLAs to their future offspring. So if we do phenotyping on the parents, then we could reasonably assume what type of HLAs they will be giving to their offspring. So let's briefly look at this chart real quick. So if we are doing phenotypes of our parents per se, all right. So we have our mother's haplotypes, and we're just doing HLA A and B types right now, just to simplify it. So our mother, the mother's haplotypes would be A9 and B27. Remember, they inherit them as a set or a haplotype. And also A2 and B14. The father's HLA types would be A1 and B8, and then A28 and B5. So there could only be 4 possible combinations with these different haplotypes. All right, so these are the separate or the different haplotypes that one child could inherit. So they have a 25% chance at getting one of these inherited haplotypes from their parents. So here's a quick chart. Let's go ahead and fill this out. So we know the mother and father's haplotypes. I want you to be able to determine the possible child's haplotype. So the four possible child haplotypes that they will inherit from their parents. Go ahead and fill that in or write it down on a piece of paper, and we'll go over them shortly. Okay, so we have child 1. Alright, so they could possibly inherit the mother's A9, B14, CW4, and the father's A1, B8, CW1. Or they can inherit the mother's A9, B14, CW4, and the father's A10, B18, and CW5. Another possibility would that they would inherit the mother's A2, B27, and what is significant about B27, that would be ankylosing spondylitis, and CW3, and the father's A1, B8, CW1, or another option would be the mother's A2, B27, CW3, and the father's A10, B18, CW5. Now, one way to simplify this would be to take the mother's haplotype, the first one, the A9, B14, and CW4, and put it down on the first two children. Then take each of the father's haplotypes and put it right behind the mother's. Alright, so that's basically it. So we have remember you have to have one from the mother and one from the father. On child's 3 and 4, then whatever haplotype you didn't use in the top 2 rows of child 1 and 2, you'll put down in 3 and 4. So that would be A2, B27, and CW3. And then you'd fill in the father's haplotype for those. So remember that we're filling in the child's haplotypes that they could potentially get from the parents, and remember that the child will inherit one set of haplotype from the mother and one from the father. So we could use HLA phenotypes for to help determine the potential child, what the child will inherit from their parents, or we can determine who the mother of the child was, as long as we have the father and the child's phenotypes, or determine who the father is if we have the mother and child's phenotypes. So we can do paternity testing to determine the potential father, or maternity for potential mother, whichever case it may be. before you start clicking to go to the next slide, there will be some circles and lines drawn on this slide. So let me go ahead and get started. So we are doing a paternity test right now. We're trying to determine the potential father to see if this potential father is actually the father of this child. So the first thing we want to do is, and after I get done speaking, you'll see these circles pop up. So first we want to identify the haplotype that the mother gave to the child, because we know that this mother is the mother of this particular child. So when you click through, you will see that I have circled the mother's haplotype that she gave to her child, which is A9, B27, CW3, and DR2. So we know for a fact that A, this is the mother of this child, and B, that she gave this child the HLA A9, B27, CW3, and DR2. Now, what is left over, okay, so we've already used those and we can't use those again. So what is the other haplotype that the mother has? So that would be everything left over. So that would be A2, B14, CW4, and DR5. And we would write that out to the side of the mother's haplotype. And the same for the child. So the child's other haplotype would be A28, B5, CW5, and DR4. And this is just showing you that we're doing the leftovers. So after we figured out the mother's haplotype and what she gave to the child, then the leftovers would be written as the second haplotype for the mother and the child. So like I said before, it would be A2, B14, CW4, and DR5. I'm sorry, that should actually be CW3. And for the child, it would be A28, B5, CW5, and DR4. So now we know the child's haplotype, okay? Both sets. So we know that the mother gave this child A9, B27, CW3, and DR2. So the other haplotype must have come from the child's father. So now we know it's A28, B5, CW5, and DR4. So now let's look at the potential father's haplotype. We have an A28, all right, we have a B5, we have a CW5, and we have a DR4. So this child must be the child of this potential father because we have a match. So here's another example. So I want you to write down the mother's haplotypes and determine if this is the father of the child. Please review this lecture. You will have paternity testing on the exams, so please learn how to do this. If you have any questions about this, please get in contact with me. I'll be happy to answer any questions that you may have.